Okay, hi everyone. I have the warm up from last week that you can work on for now. We'll just wait for some more people to stream in. If they do, it's Thanksgiving, so I'm not sure if we'll. But we'll work on this warm up for like 2 or 3 more minutes. If you have answers, you can send them to me in private chat. Okay, so to everyone who just joined, we'll work on the warm up for a few minutes as we wait for more people to come in, and then we'll get started on the new content. Okay, I will go over the answers now. I saw a lot of good answers. So for the first question, what are the reactants and products of photosynthesis? Our main reactants are carbon dioxide and water, and our products are glucose and oxygen. Number two, the waxy layer that covers the epidermis and help prevent water loss is called the cuticle. In vascular plants, the tissue that transports water and minerals from the roots to the leaves is called the xylem. And for the multiple choice, what do we call the tiny pores on the underside of a leaf that allow gas exchange? The answer is C, the stomata. Okay, so that brings us to our new topic for today, which is inheritance and variation. So we'll go over genetic material, what variation is, how inheritance works, and then Mendelian genetics. So starting off with genetic material. So firstly, what are genes? So genes are the basic unit of heredity. So they are segments of DNA that carry instructions for making proteins. And these proteins are very important for the structure and function of the body's various tissues and organs. Genes are located on chromosomes within the nucleus of the cell. If you remember, like the nucleus is the control center of the cell, and each gene provides the instructions for a specific trait. So for example, for your eye color or for your blood type. Different versions of the same gene. So for example, like a gene for blue eyes versus a gene for brown eyes are called alleles. And these variations can result in different traits. Okay, so what are chromosomes? Chromosomes are these long thread-like structures made up of DNA and proteins, and each chromosome is made up of many genes. The DNA in chromosomes is very tightly coiled and packed to fit within the nucleus, and this coiling is important for efficient storage and organization of our chromosomes. The importance of chromosomes or the role is to ensure that DNA is accurately copied and distributed in the process of cell division or the process of making new cells from existing cells. They help us pass genetic information from one generation to the next generation. In humans and many other organisms, chromosomes come in pairs. So one set of chromosomes is inherited from each parent. And then looking broader, DNA. So what is DNA? The full name for DNA is deoxyribonucleic acid, as you can see spelled on the top. It is the molecule that carries all the genetic instructions for life and teaches cells how to build and maintain an organism. So we can see the structure of DNA kind of here. in this simple diagram, it's a double helix, which means it's kind of like a twisted ladder, and the rungs of the ladder are made up of pairs of nucleotides. So there are 4 nucleotides in DNA, which are represented by the letters A, T, C, and G. So A is adenine, T is thymine, G is guanine, and C is cytosine. And there are these rules of base pairing, which means A will always be paired with T, and C will always be paired with G. So you can kind of see in this diagram here, all the red ones are composed of A and T together, and all the blue ones are composed of G and C together. So before a cell divides, it will make a copy of its DNA so that each new cell gets a complete set of instructions. So we can see a real image of DNA here of that double helix structure. It's like very, very blurry because it's very, very small. But below you can see like the model that's supposed to make that helix structure a little bit more clear. And you can see also on the right, like kind of like the squiggles of the helix. So going back to chromosomes, we talked about how they are in pairs. So when you look at humans, we have 46 chromosomes, which are 23 pairs of chromosomes. The first 22 pairs of chromosomes are autosomes. So these contain genes that determine most of the body's characteristics and functions. Like if you think of auto, that normally has something to do with the body. And then the 23rd pair of chromosomes are your sex chromosomes, so X and Y, which determine the gender of an individual. So females have 2 X chromosomes and males have 1 X and 1 Y chromosome. Okay, so a summary. Genes are segments of DNA that code for proteins and determine your traits. And then we have DNA, which is a molecule carrying genetic instructions in this double helix structure. And then we have chromosomes, which are structures made up of DNA and proteins, contain many genes within them, and all humans have 20, or not all humans, most humans have 23 pairs of chromosomes. So kind of a diagram to put that into perspective. We start with our single nucleotide, which is paired with its base pair. So it might be A with T or C with G, and that composes our double helix DNA. Each section of DNA is a gene that codes for a specific trait. And then we have chromosomes, which are made up of all this tightly coiled DNA, and then is stored within the nucleus of a cell. And then we are made up of many, many, many cells. Okay, so looking into inheritance now. There are 2 types of traits, which are dominant traits and recessive traits. So dominant traits are genetic characteristics that are visibly expressed, even if an individual has only 1 copy of the dominant allele. So allele, I mentioned, is like variation of a gene. It's represented by a capital letter. So for example, capital B for brown eyes. Recessive traits are genetic characteristics that are only expressed if the individual inherits 2 copies of the recessive allele, and they're represented by a lowercase letter. So for example, lowercase b for blue eyes. So looking at some examples of dominant versus recessive traits. So if you have 2 recessive alleles, you'll have the recessive trait, but if you only have 1 allele for the recessive trait and 1 for the dominant one, the dominant trait will mask the recessive one and you won't have it, which makes it less common. So we can see like dimples are dominant, free earlobes, being able to roll your tongue, etc. So the term for having 2 different alleles of a gene is heterozygous. So going back to our example of capital B for brown eyes and lowercase b for blue eyes, a heterozygous pair would be capital B, lowercase b. So this means that you have 1 dominant trait and 1 recessive trait. We know that you need 2 recessive traits for them to actually be expressed or visible in the individual. So someone with capital B, lowercase b would have brown eyes. Conversely, homozygous is having the same alleles of the gene. So either 2 dominant alleles or 2 recessive alleles. So in our case, either 2 capital Bs or 2 lowercase b's. So the pictures show the combinations that you'd need to get either brown or blue eyes. They used E as their letter, but it's the same kind of meaning. So as long as you have at least 1 dominant allele, you will have brown eyes. To have blue eyes, you need both of the recessive alleles. So what we can use to predict the probability of inheriting certain traits is a Punnett square. So it's this kind of grid that shows the possible combinations of alleles from two parents. So how we use it is we write the alleles of one parent along the top and then the alleles from the other parent along the side. And then we fill in the squares by combining the alleles from each parent. So in this case, capital B, capital B, capital B, lowercase b, etc. And then we'd analyze the resulting genotypes or genetic makeup to determine the possibility of each phenotype or visible trait. So these are all individually genotypes, but when we look at the phenotype, that would be either brown eyes or blue eyes. So for example, if we're crossing two heterozygous, so they have different alleles, brown-eyed parents, so capital B, lowercase b, crossed with capital B, lowercase b, we have our three possible genotypes here. So two capital Bs, one capital B and one lowercase b, and then two lowercase b's. So our phenotype ratio is 3 brown-eyed to 1 blue-eyed, because we know that as long as you have one capital B, you will have brown eyes. So another example is crossing capital B with capital B lowercase b. In this case, you can see that every offspring will have at least one dominant allele. So our phenotypic ratio in this case is 4 brown eyes to 0 blue eyes. Green eyes, someone asked what about green eyes. Green eyes are also a recessive trait, so you could like sub that in for blue eyes in this situation. Okay, so now looking at variation. So what is variation? Variation is differences within a species. So differences in traits among individuals all of the same species. For example, different fur colors in dogs. They come in black, brown, white, spotted. And you can see some examples of variation here in these shells, the beaks of these birds, again, dog fur color. And the importance of variation is to provide adaptation. So it enables populations to adapt to changes in their environment. So once conditions change, for example, shifts in climate or new predators introduced or diseases, organisms with beneficial genetic traits can survive those and then reproduce more successfully and pass those advantageous traits on to the next generation. We kind of talked about this in ecology and ecosystems. They also provide disease resistance. So if we had a population where all individuals were genetically identical, that means a single disease or environmental change could just wipe out the whole population because they're all the same. So having diversity in genes provides a buffer against these kind of events occurring. Biodiversity health. So healthy ecosystems rely on the diversity of organisms to maintain balance. And within a species, genetic variation ensures that some individuals can survive and thrive under like a varying amount of conditions to maintain ecological stability. Agricultural benefits. In farming and agriculture, genetic variation is used to breed crops that are more resilient to pests, diseases, and different environmental conditions. And then evolutionary potential. Without variation, there wouldn't be that selection and passing on of advantageous traits, and then there would be no evolution because there's nothing to change or select for and against. So looking at some environmental factors affecting phenotypes. So again, phenotype is like the visual expression of a genotype. So we can say like blue eyes is a phenotype. So some things that can affect phenotypes are temperature. So certain traits like coat color in animals can be influenced by temperature. Looking at this picture here, we have a Siamese cat. The color of the fur in Siamese cats is darker in cooler areas of the body due to them having these temperature-sensitive proteins that affect the production of pigment. Also, light. Exposure to light can affect plant growth. We talked about phototropism previously, and also flowering times. In animals, light can influence behaviors like mating, migration, or the development of certain features like coloration in birds, and then altitudes. So organisms living at higher altitudes often exhibit phenotypic adaptations to cope with the lower oxygen levels at higher altitudes. So We know that humans that are born and raised at high altitudes can develop larger lung capacities and more red blood cells. I've also heard of like a certain population having better capacity to like swim underwater for a longer period of time. So it's kind of similar situation, as well as chemicals, nutrition, stress, social environment, etc. So there's a lot of factors that can affect phenotype. Okay, so looking at mistakes in DNA, mutations are changes in the DNA sequence that can lead to new traits. They can occur either spontaneously or due to environmental factors like radiation or chemicals, and mutations can be beneficial, harmful, or just have no impact at all. So we have three kinds of mutations. The first one we have is a deletion. So pretty self-explanatory. It occurs when one or more nucleotides, those building blocks of DNA, are removed from the DNA sequence. Then we have substitution. So when one nucleotide in the DNA sequence is replaced by another. So for example, like originally we had A, but then we replace it with G. That would be substitution. And then we have insertion. So this occurs when one or more nucleotides are added to the DNA sequence. Okay. Next, looking at Mendelian genetics. So Mendelian genetics comes from Gregor Mendel. He was an Austrian monk born in 1822 and was known as the father of genetics for his work in understanding inheritance. So he did some experiments to understand how traits are passed from parents to their offspring. So he worked primarily with pea plants. You can see a picture here. He chose pea plants because they have easily observable traits. So like what their flower color is or what the shape of their seeds are. And they can also self-pollinate and also be cross-pollinated. So what he did is cross-pollinate pea plants with different traits to see what the outcomes would be over multiple generations. And then he conducted these experiments on thousands and thousands of pea plants to make sure that his results were accurate and reliable. So what he found out from crossing all these pea plants and examining their offspring were that traits are inherited in predictable patterns, and each trait is controlled by certain factors, which we now know are genes that occur in pairs. So he came up with a few laws of inheritance. So firstly, the law of segregation. So what this means is that each individual has two alleles from each gene. So one inherited from each parent. Like we talked about the eye color thing before, everyone has one allele from each parent that ultimately determines what their eye color will be depending on how many dominant or recessive alleles they have. So in terms of pea plants, for a gene determining flower color in pea plants, one plant might have two alleles for purple flowers. So like double capital P, two alleles for white flowers, two lowercase p's, or one allele for each. So one capital P and one lowercase p. These alleles will separate during formation of the offspring. So that's where like the segregation comes in. So a plant with capital P, lowercase p can produce gametes with either dominant purple or recessive white. So if that sounds kind of confusing, like going back to our Punnett squares, this is kind of demonstrating like the law of segregation is that the two alleles that each parent have like will independently determine a possible circumstance, if that kind of makes sense. So the alleles from each parent are independently segregated when determining like possible future traits. So we consider their dominant one and also the recessive one separately when we determine the probability of different phenotypes occurring. Okay, so this is also a kind of diagram that basically demonstrates the same thing as a Punnett square, but we can see how we look at like the separate alleles of the father and the mother, both of which they're called carrier parents because they have these recessive recessive alleles present. So in this case, the recessive one is the red lowercase r. And we can see that although they are unaffected, as they do have one dominant R also, when we cross all the possibilities of the children that they could have, there is a 1 in 4 chance that they will have a child with 2 recessive alleles. So the law of segregation basically just tells us that we need to look at all these possibilities of their alleles separately. His second law was the law of independent assortment. So genes for different traits can segregate independently, and the inheritance of one trait does not affect the inheritance of another trait. So the alleles for different genes are distributed to gametes independently, leading to genetic variation. So when he was doing this with pea plants, he proved this by looking at 2 traits, so seed shape and seed color. The seeds of pea plants can either be round or wrinkled, and the seed color can either be yellow or green. So he found that the inheritance of seed shape does not have anything to do with the inheritance of seed color. So for example, if capital R, like the dominant trait is a round seed and the recessive trait is a wrinkled seed, dominant color is yellow and recessive color is green, a plant that is heterozygous for shape and color can produce gametes with any combination of these alleles. So that means they can have like RY, capital R, capital Y, capital R, lowercase y, lowercase r, capital Y, lowercase r, lowercase y, like you can see here. So that means if we are crossing that plant, for example, that had heterozygous for both seed, shape, and color, this is how we would write it out as our Punnett square, because any of these possibilities are possible and they're independent from each other. So having a round seed doesn't have anything to do with being yellow, and having like a green seed has nothing to do with being wrinkled, kind of like that. So a lot of possibilities to consider. This is a pretty complex Punnett square. Polygenic inheritance. So what polygenic inheritance is a type of inheritance where multiple genes, which are often called polygenes, contribute to the expression of a single trait. So instead of being controlled by a single gene with 2 possible alleles, like we were looking at with those Punnett squares and Mendelian inheritance, polygenic traits are influenced by several genes, and each of those genes has like a small additive effect on the phenotype. So traits controlled by polygenic inheritance usually show a range of continuous variation rather than distinct categories. So for example, when we were looking at Mendelian inheritance, it's either you have blue eyes or you have brown eyes. You don't have like half blue, half brown eyes, although I'm sure that is possible, just not common. So examples of these polygenically inherited traits include height, skin color, weight, that typically follow a bell curve of distribution that is more of like a scale. Like it's not like you're either 6 feet or 5 feet. You can be like any range of heights around in between there. And most individuals will have an intermediate expression of that trait, and fewer individuals will exhibit extreme values of the trait. So, for example, I think the average height for women is around like 5'5". Most women will be around there. And then it kind of, the probability of being either super tall or super short kind of decreases as you deviate from that 5'5". So looking at those additive effects, in polygenic inheritance, each gene contributes a small amount to the overall trait. So the more positive alleles that an individual will inherit, the more pronounced the trait becomes. So for example, looking at the height of a person, it's determined by the cumulative effect of several genes, each contributing to the height of a person. So unlike traits determined from a single gene, polygenic traits do not follow a clear dominant recessive pattern. Instead, the effect of each gene adds on to the one of the previous ones, and then the final phenotype or expression of the trait comes from the combined influence of all the genes involved. Polygenic traits are also often influenced by environmental factors, which makes them a little bit more complex to predict. So for example, your height is determined by your genes, but is also significantly affected by your nutrition during development. So looking at that kind of bell curve that we talked about, where the majority of the outcomes will fall somewhere around the median, and then becomes less likely to be at those extremes. So in this case, we're looking at corns, that have 3 genes involved in the situation with either a pure white or pure red. You can see that pure red is very rare as well as pure white, and it's more likely to have some intermediate combination of those genes. Same with height, like I was talking about earlier, most people will be around the average height, and then you'll see less people who are super tall or super short. Okay, so finally looking at some modern genetics techniques. So genetic engineering involves directly altering an organism's DNA to modify or enhance specific traits. So we can do this by adding, removing, or altering genes to produce desirable characteristics or to study the function of genes. Scientists can also transfer genes from one species to another that produces organisms with traits that are not naturally found in them. So for example, that could be something like introducing a gene from a cold-resistant fish into tomatoes, and that way the tomato plant can survive the frost. So looking at some specific techniques, firstly, CRISPR, which stands for clustered, regularly interspaced, short palindromic repeats. So a pretty long name, but essentially how it works is an enzyme called Cas9 acts as these molecular scissors to cut DNA at specific locations guided by a custom RNA sequence. So once that DNA is cut, scientists can add, delete, or modify material at that spot. So you can see in this diagram, we have that Cas9 like kind of scissors that makes a little cut here, and we can either add something, like in this case, or remove something, remove the genes. So this has applications in treating genetic diseases, improving crops, and also just studying how genes function. Then we have GMOs, which are genetically modified organisms. And these are organisms that have had their genomes altered using genetic engineering techniques to produce new traits. So typically involves transferring genes from one species into another to provide specific benefits, like that resistance to the cold we talked about with like the tomatoes and the fish. They are widely used in agriculture to create crops that are more resistant to pests, diseases, or herbicides, and are also used in medicine to produce things like insulin or vaccines. Then we have, lastly, selective breeding, which is also known as artificial selection. It's a traditional method that doesn't involve all this technology, where humans will breed organisms with desirable traits to enhance these characteristics in future generations. So unlike genetic engineering, this method relies on natural reproduction and the selection of specific traits over many, many generations. So the limitation with this is that it is slower and less precise than genetic engineering because it depends on naturally occurring genetic variation. So this can be something like, in this case, we have these trees or plants or something. If we want them all to be taller, we will like cut down all the short ones, let the tall ones grow, reproduce, and so on until we have this second population of tall trees. Or it can be things like breeding dogs for specific traits like their size or their attitude. So a review of everything we went over today. Firstly, genes are segments of DNA with instructions to make proteins. Then DNA are molecules carrying the genetic instructions for life. And then we have chromosomes, which are made up of DNA and also proteins. And humans have 23 pairs or 46 chromosomes. Then we looked at traits and how they can be dominant or recessive. Heterozygous is a pair of one dominant and one recessive, and homozygous alleles are either dominant and dominant or recessive and recessive pairs. We can use Punnett squares to predict possible genotypes and the probability of each occurring phenotype. Variation is genetic difference. We looked at why variation is beneficial in populations. Then we looked at mutations, which are DNA changes that can be beneficial, harmful, or neutral. There are 3 types: deletion, substitution, insertion, fairly straightforward. Then we looked at Gregor Mendel and Mendelian genetics and his law of segregation, which is that every individual has 2 alleles, one from each parent. Then the law of independent assortment. So genes for different traits segregate independently. They don't have anything to do with each other. And then we talked about polygenic inheritance. So inheritance of traits that is controlled by multiple genes instead of like the normal one from each parent. And then we looked at some modern genetic techniques. So does anyone have anything they want to look over again before we move on to the booklet? Yeah, so someone asked about like the additive effects. So going back to the polygenic inheritance, what additive effects means that each gene contributes a little bit to the overall trait. So we talked about height. Height is determined by the cumulative effect of several genes that ultimately determine the height of a person and doesn't follow that either like dominant or recessive pattern. So basically the effects of all these genes add up to each other to determine what your height will be. Any other questions? Also, next week we will be looking at evolution. Okay, if not, I will get the booklet set up. And if you guys have any ideas for like the game mode that you want to play. Someone said crypto. Anyone have, okay, someone said not crypto. Does anyone want to break the tie on that? Okay, someone else said not crypto. So if not crypto, then what? Here are your options. We have Candy Quest, Fishing Frenzy, Crypto Hack, Tower Defense 2, Monster Brawl, Deceptive Dinos, Battle Royale, Tower Defense, Cafe, Factory, Racing, Book Rush, and Classic. Does anyone have any preference? Candy Quest, Battle Royale. If no one objects, I think we can do that, maybe Battle Royale, because we haven't done that one before. We also haven't done Factory before. Battle Royale, okay. I see 2 votes for that. We will do that this week. Do you guys want to do it solo or in teams? Team? Okay. Okay, the join link is in the chat for anyone that needs it. The code is 760289. Okay, we will get started unless anyone's like really struggling to get in. Okay. Thrifty Toucans. Okay, I didn't see the other team. Thrifty Toucans versus Fish or something. Okay, 50% of people got this one right. test results. The fiery fish won. Okay. 60% of people got this one right, and the result is that the fiery fish won once again. Hey, 40% of people got this one right. And the winner is once again the fiery fish. Okay, and the winner for this round is the once again the fiery fish. Okay, I think the thrifty toucans are like on their last life, so lock in on this one. Okay, only one person got this one right. And the result is that the thrifty toucans have lost. Okay. So that means the fiery fish are in first place. We still have 7 minutes left. We can do like another game mode quickly if anyone has a suggestion. Candy Quest. Okay, let's do Candy Quest. The join link is in chat. The code is 1777550 if you need. Okay, I will get started, but late joining is allowed. Okay, we have 4 minutes remaining, and in first place we have Didia. The final few seconds, we have Joy in first place. Okay, final standings. Didia in third, Joy in second, and I'm hacking in first place. Thank you, everyone. That is all for today. I hope you guys have a good Thanksgiving. Okay, bye, everyone. Bye, everyone.